Punnett squares, dominant versus recessive, those classic 3:1 and 9, 3, 3. 1 ratios, do they actually make sense, or just feel like memorization? For the MCAT, Mendelian genetics is high yield, and understanding the logic behind inheritance patterns can turn guesswork into confidence. In this video, we'll break down how traits are passed, how to predict outcomes quickly, and how to spot the traps test writers love, so you can walk into test day ready to think, not memorize. Mendel's law of segregation states that allele pairs separate during gamete formation, so each gamete carries only one allele for each gene. Think of a diploid organism with genotype AA. During meiosis I, homologous chromosomes separate, and by the end of meiosis, each gamete receives either A or A, never both. A helpful mnemonic is separate in meiosis, single in sperm or egg. On the MCAT, this shows up in predicting offspring ratios from crosses. For example, crossing 2 heterozygotes, AA times AA, yields a 1:2. 1 genotypic ratio, AA, 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 and a 3, 1 phenotypic ratio, if A is dominant. You should be comfortable drawing a Punnett square quickly or using probability rules. The chance of AA is 1/4 because each parent contributes A with probability 1/2. So 1 divided by 2 times 1 divided by 2 equals 1 divided by 4. This law also explains why recessive traits can skip generations. Heterozygous carriers, AA, are phenotypically normal, but can pass on the recessive allele. Connect this to meiosis mechanics. Errors in segregation can lead to non-disjunction, which is more often tested with chromosomal abnormalities like trisomy 21. Another common twist is test cross logic. If you have a dominant phenotype with unknown genotype, A, cross with a homozygous recessive chrom A, if any offspring show the recessive phenotype, the unknown parent must be heterozygous. Finally, remember that segregation assumes no linkage and no meiotic drive. When those assumptions are violated, ratios deviate from classic Mendelian expectations, which is a clue in passage-based questions. Mendel's law of independent assortment states that alleles of different genes are sought independently during gamete formation, when the genes are unlinked. In practical terms, if you track 2 genes, say A, and B, on different chromosomes, the gametes from an AAB individual are AB, AB, and AB in equal proportions of 1/4 each. A mnemonic is different chromosomes, different fates. The classic MCAT application is the dihybrid cross AABB times AAB, which yields the 9, 3, 3. 1 phenotypic ratio under complete dominance, 9 with both dominant traits, 3 with only A dominant, 3 with only B dominant, and 1 with both recessive. You can derive this either with a 4 times 4 Punnett square or by multiplying independent monohybrid probabilities. 3/4 dominant for A times 3/4 dominant for B gives 9/16, and so on. The exam often tests whether you recognize when independent assortment does not apply specifically, when genes are linked on the same chromosome. Linkage reduces recombinant gametes and skews expected ratios, which ties into recombination frequency and genetic mapping, a higher yield follow-up topic. Another common variation is asking for the probability of a specific genotype or phenotype across multiple children. Here, use independence across both genes and offspring, multiplying probabilities accordingly. Also connect to meiosis, i.e. metaphase alignment, where homologous chromosome pairs orient randomly, underpinning independent assortment. When you see unexpected ratios in a passage, consider linkage, epistasis, or small sample size as explanations, rather than abandoning Mendelian logic entirely. Dominant and recessive inheritance patterns determine how alleles translate into phenotypes, with complete dominance producing classic 3:1 ratios in monohybrid crosses. In complete dominance, one allele masks the other in heterozygotes, so AA looks like AA. A quick mnemonic is dominant displays, recessive requires a pair. On the MCAT, you'll frequently be asked to infer genotypes from phenotypes and pedigree data. If two unaffected parents have an affected child for a recessive disease, both parents must be carriers. Conversely, a dominant disorder typically appears in every generation, though incomplete penetrance can complicate this. Be ready to distinguish autosomal versus X-linked patterns. X-linked recessive traits appear more often in males because they have only one X chromosome. Carrier females can pass the trait to sons. For autosomal dominant traits, expect vertical transmission affecting both sexes equally. Another testable angle is predicting outcomes of crosses involving homozygous dominant, AA, heterozygous AA, and homozygous recessive AA individuals, especially when asked for probabilities across multiple offspring. Use binomial reasoning if needed. Real-world examples include cystic fibrosis, autosomal recessive, and Huntington disease, autosomal dominant. Connect this to molecular biology. Dominance often arises because one functional allele produces enough protein, whereas recessive phenotypes reflect loss of function. Finally, watch for wording like true breeding, which signals homozygosity, and for trick answers that ignore carriers when calculating risk. Punnett squares and probability rules are tools to predict genotypic and phenotypic ratios in genetic crosses. While Punnett squares provide a visual method, the MCAT rewards speed, so mastering probability is key. Use the multiplication rule for independent events probability of both A and B is PA times PB, and the addition rule for mutually exclusive events, PA or B, is PA plus PB. For example, in AA times AA, the probability of a heterozygous child is 1/2. For two children both being heterozygous, multiply 1 divided by 2 times 1 divided by 2 equals 1 divided by 4. For at least one questions, it's often faster to compute the complement, 1 minus the probability of none. A classic example is the chance that at least one of 3 children is affected by a recessive disease when both parents are carriers. Compute 1 3/4 3. In dihybrid scenarios, treat each gene separately if they are sought independently. Then multiply outcomes. Another high-yield trick is the forked line method, which replaces large Punnett squares by branching probabilities for each gene and then combining paths. Be cautious with sex-linked traits. Probabilities differ by sex, so sometimes you must condition on whether the child is male or female. Also, remember that observed ratios may deviate from expected ones in small samples. Many passages include chi-square reasoning qualitatively, asking whether data fit Mendelian expectations. Connecting to statistics and experimental design can help you interpret these deviations correctly under time pressure. Non-Mendelian patterns such as incomplete dominance, codominance, and epistasis modify classic Mendelian ratios and must be recognized on the MCAT. In incomplete dominance, the heterozygote shows an intermediate phenotype, think red and white flowers producing pink, yielding a 1:2:1 phenotypic ratio that mirrors the genotypic ratio. In codominance, both alleles are fully expressed, as in the AB blood type, where both A and B antigens are present. Here, the phenotype directly reflects both alleles rather than blending. A mnemonic is incomplete blends, codominant both show. Epistasis occurs when one gene masks or modifies the expression of another, producing altered ratios like 9:3, 4, or 12:3, 1. For example, in Labrador coat color, one gene controls pigment production, and can mask the color gene entirely. The MCAT often tests your ability to identify these patterns from ratios or descriptions, rather than naming them outright. Connect this to biochemical pathways. Epistasis frequently reflects upstream enzymes, controlling whether a downstream product can even be made. Also be aware of pleiotropy, where one gene affects multiple traits, and of penetrance and expressivity, which explain why phenotypes may not perfectly match genotypes in pedigrees. Finally, link back to molecular genetics. Different mutations can alter protein function in ways that produce these inheritance patterns. So recognizing the pattern helps you infer the underlying mechanism and choose the correct prediction. Under exam conditions,